Okay, so thank you, Alison, again. And I would say, in the interest of time, we move on to the next presentation, which will be given by Joost van Griethuysen, who will be talking about PyRadiomics. Joost is the resident in radiology, and he's very interested in software, as you'll see in a few moments. Right, Joost, the stage is yours. Welcome.
Thank you for the kind introduction. I hope everyone can see my screen now, so I have nothing to disclose. And my talk will actually be about PyRadiomics, and maybe it seems a bit strange because indeed I have studied medicine as my main area of interest, but then I started with a PhD on artificial intelligence on medical imaging and with specific attention to imaging in rectal cancer. And as part of this, I joined up with the team developing PyRadiomics. But before I go into this specific package itself, I wanted to have a little recap on what is radiomics, because I'm not sure if it's as clear to everyone here. So in essence, radiomics is the objective quantification of tissue heterogeneity, but usually we say that it's a way to capture the radiographic phenotype, so how an area of interest looks on an image in a way that a computer can understand. And we do that by extracting a large number of predefined mathematical formulas, which we typically name the radiomic features. And these are generally around, well, more than 1,000 features are generally extracted from a single image. And the trick then is to combine the best of these features to join them into a model which ultimately predicts the outcome. And actually, this is what is known as a data-driven analysis. So we don't really start with a hypothesis, but we have we have this many features. Are any of them correlated with our outcome of interest? And the model itself is then, of course, needs to go hypothesis testing. Which is this model a good model? Interestingly enough, we can also compare how a computer using radiomics looks at an image to how a radiologist does it. So it starts at the first part. So you have your image, and then you need to do some sort of pre-processing. So for the radiologist, he will adjust the window and level so the contrast really emphasizes the area he's looking at. For radiomics, these processes entail stuff like normalization or resampling to make the images more comparable to one another. Next part is that the lesion, of course, needs to be located. So for the radiologist, he will go looking and scrolling through the images until he finds this lesion. And for radiomics, there we have to help the computer a bit by providing it with a segmentation, which specifically signifies which is the area of interest, which will generally be a tumor or a metastasis or something like that. Next part is, of course, the description of the lesion. So this is not a classification, but merely the description. So for radiologists, he will be using qualitative features, which are like the morphology. It's a very round or very spiculated tumor, generally the size. So for radiomics, this is the feature extraction, where we extract these quantitative features, which are generally related to stuff like histograms, so which gray values are present in the image. And also the shape. So what is the shape of the image? So the roundness, but also the volume and the maximum size. And the third group, the biggest group, are the texture features, which describe the relationship between the gray values and their spatial location within the area of interest. The final step is to classify the lesion. For the radiologist, he will be using standards like the TNM stage, which are based on his description of the image and comes out with his classification. And for the radiomics, this is where the classifier is trained and then applied, which will generally result in a probability of the desired outcome. This stepwise approach, if you take these first three steps, these are generally considered the feature extraction. The fourth part is the analysis and modeling, which will be a bit outside of the scope of this talk. And I'm quite sure there are many good talks about how to analyze and model this big data, but I want to focus a bit on this first part, the feature extraction. And for that, I want to start with already this first part, so this pre-processing. So we can't just take an image and throw a bunch of mathematical formulas at it and hope for the best. So we first need to do some steps. And one of the most known steps is actually image discretization. So that means that for the texture features, you can't use the original gray values. You need to make, because in general, if you take, for example, a CT, there are 5,000 possible different gray values. And if you use that many gray values, your texture features will mean nothing. The probabilities of each individual gray value are simply too low. So we need to reduce the number of gray values, and this process is known as discretization. And what you actually do is you bin gray values together in a histogram and then take the histogram bin numbers as the discretized gray values. Generally, there are multiple ways of doing this. The most two popular ones are either to use a fixed bin width. So for example, you take a fixed number of gray values, you bin them together. So for example, the gray value 0 to 100 gets assigned to bin 1, 101 to 200 to bin 2, and so forth. And the other one is a fixed bin count. And this means that you take the entire range, say your region of interest has 300 different gray values, and you say, okay, I want to bin them together in about 50 bins. So then you calculate and you have 15 gray values per bin, and then you discretize your image. The bin count is easiest because you just you are ensured you always have an adequate number of bins. However, this also means that your gray value meaning is actually dependent on the range. And the best way to describe that is that if you take one image that is very heterogeneous and has a large range of gray values, then each bin will contain a significant amount of gray values and be something akin to what also a radiologist will consider a reasonable difference in gray value. However, if you have a very homogeneous image, say if you have an image with only 100 different gray values and you try to make 100 bins out of it, that means that each single gray value will get assigned to its own bin. And especially in CT, if you ask any radiologist, well, would you consider a difference of 1 Hounsfield unit to be a realistic difference? He will say, no, of course not. So this is a problem that you do need to pay attention on it. So the fixed bin width has the problem that sometimes you get too many or too few bins, but at least it has the added the benefit of having the same meaning of a gray value. And this is actually also what you see back in research. So this is actually a quite old study from 2015 already, where they compared these relative and absolute, so the fixed bin count and the fixed bin width with each other for discretization, and actually say, okay, well, if you use a fixed bin count, your reproducibility of your features is a lot lower. And also more recent research from 2019 also shows this in the characterization of MRI features, that you really need to go for a fixed bin width if you want to have good reproducible features. Another problem is that images are often of different pixel sizes. And for the radiomics to work, you need to have at least some homogeneous pixel sizes. And we can easily fix that by resampling, but then you get the problem of, okay, how do you fit this new grid? So do you say, okay, I want to have my corners aligned or the centers aligned? And for both approaches, they are valid resampling. Only you do need to realize that these will slightly differ in the results of the resample pixel values, and therefore also the extracted features. Fortunately, in recent years, there's been a new initiative called the Imaging Biomarker Standardization Initiative, which aimed for creating a description of standardized feature extraction, which actually, they started out with 24 different teams. And through many meetings, we tried to get some sort of standard on how one should perform a radiomics feature extraction. And what we actually saw is during these meetings, the concordance between the different feature packages actually increased dramatically. If you look at here at the phase 1, where we're just looking at the feature formulas without any pre-processing, at the start, there was over 75% of packages of features that were discordant between the teams. And at the end, they were all at least partly concordant. And the same goes for phase 2, where we also added a limited form of pre-processing. Finally, also in retest cases, many features show good reproducibility, but again, this paper showed that the pre-processing parameters are very important, making the features very sensitive to them. And in some cases, that even normalizing the images doesn't really solve this problem. Finally, also in multi-center data, in this case where all the pre-processing steps were kept as the same as possible, we saw that the largest source of variance actually was the acquisition. So what scanner and what hardware was used, but also the acquisition protocol. Again, a point of interest, but which you can't really change if you're using retrospective data. For the segmentation part, also it's outside the scope of the PyRadiomics project, but again, it has been known that whole volume segmentations lead to more reproducible features than single slice. And finally, the software implementation, which is the part of Radiomics, showed that the variance is mainly in the higher order features, which is partly because these features are that much more complex, but also because these features generally require a bit more pre-processing than simple first order features like shape and histogram features. So the challenges. So the first part, of course, is the software, which for several years ago, it was mainly in-house developed and not shared. So papers were not that much reproducible. And the second thing, also the feature definitions, so no gold standard. So there are several features that have predefined mathematical standards, but again, the implementations differ slightly, leading to very different results. And as I've shown, also the pre-processing is a very large influence, but still often underreported. Outside of these things, we also have to take into effect the image variation, so differences in segmentations, but also scanner and protocol differences and day-to-day acquisition noise. These parts all highlight that the standardized extraction, but also a very detailed description of methods is needed, and sad to say, is still often lacking in papers published today. So our initiative here was PyRadiomics, and our thought here was, well, we can try and describe what people should do, or we can try and build something people can use so they don't have to reinvent the wheel themselves. And this led us to the development of PyRadiomics, which actually is an open source toolbox, free of use, with the only added benefit that you actually had to cite or work that we did. And we aim to actually generate a community of radiomics researchers that could share ideas and work with the same software to make the research they publish more comparable, leading to better and newer insights. So to facilitate this, so the source code, of course, is hosted on GitHub, but we also provide precompiled stuff on PyPy and Anaconda Cloud, so that even researchers that only have limited programming experience can do a full radiomics feature analysis. To help people a bit along, we've spent a lot of time working on the extensive documentation. So it's not just a description on how to use the code, but also on the background of the feature. So what does this mean? And what does if I see a value like this, well, what do I need to think about this value? And what are some of the considerations you have to do when you're setting up a research? And finally, we really aim to go with PyRadiomics, we aim to go from image to feature. So the entire pre-processing part and the application of the different features, then the filters, all packaged into one single package that is fast and easy to use, with a heavy emphasis on reproducibility. So to come back a bit on these three main parts. So the first, the source code, it's in Python, which, well, nearly any researcher nowadays has in medical imaging has heard of and uses, where we also emphasize also the readability. So you can hack something together in Python, but actually to write code that is easy to understand takes a bit more time. We are also very adamant not to reinvent the wheel. So all our image handling is done on simple ITK, which is also actively maintained and is able to read and work with a host of image formats, most of which I even don't know. And in order to improve the bit and have a feature extraction that actually is done before I'm done with my researches, we actually added some C code. So it's much more difficult to read, but we found it was really necessary because at the first full Python version of PyRadiomics, it took about a week to analyze a single data set of some 100 cases. And with the C extension, that boiled down to a couple of minutes. So, but we try to keep it as minimal as possible, because adding C code really opens up a can of worms of compiling stuff and getting it to work across platforms. And of course, the readability of our source code. And finally, also because we have the C code, we also provide the continuous integration which ensures the consistency of the calculated results, but also to automate the creation of the precompiled distributions, because we found that if people have to build PyRadiomics from source, a lot of researchers run into a lot of problems because we have this C code that we actually needed to add. So on the documentation side, so this is also auto-generated from our source code, which contains not only the feature formulas, but also detailed description on how these texture matrices actually are calculated, how to use and customize the software itself. And for example, some features we have excluded because they were mathematically identical to some other features. So on the usage side, so our main intention is that it's used by researchers who have some understanding of programming. So the main interface is a simple command line script. And for those who are a bit more having a bit more customized pipeline, it also provides a single object you can build into a pipeline to facilitate your feature extraction. And the entire configuration of PyRadiomics we've coupled into a single human readable file. So this is one, it's easy to version for researcher and reproduce their own research, but also in the interest of reproducibility, you can also share this file quite easily. However, of course, there are also, as I've said, researchers that want to do radiomics but don't have this extensive programming skills. And for them, we also implemented a graphical user interface, which is integrated with 3D Slicer, which is another open source project on medical image analysis. And well, it uses PyRadiomics in the background, but all the stuff on handling the user interface and showing the images, we let that to the great minds at Slicer to program for us. And this allows you to do a radiomics feature extraction without any required programming skills. So we also are actively still developing PyRadiomics. The main addition we've added is the so-called voxel-based feature extraction. So on the bottom slide here, you see the image with in red the line, the region of interest. And in general, the segment-based radiomics is the most known one, where for each feature you get a single value that describes the entire region of interest. But in a voxel-based feature extraction, you actually define little areas, and for each area you calculate the feature and assign the value you've calculated to the central pixel in that image, and thereby you can generate a feature map, which shows you the distribution of the feature values actually throughout the images, and allows you to create a feature map, which allows you also to predict not patient level outcomes, but also region level outcomes. Finally, another experimental extension is, of course, is Daikon. It's generally used a lot, and it has very great cross-referencing and versioning in there, but it's generally a bit more difficult to use. So what we've added in PyRadiomics now is actually a fully Daikon pipeline where you can put in Daikon images and segmentations, which will get converted, processed by PyRadiomics, and where the results are actually also stored in a Daikon SR document. So you can have a fully Daikon pipeline using PyRadiomics. This, of course, is not generally used in research today, but especially if you have clinical implementation at some point, you also need to store the image results in a way that is compatible with the current day implementations. As I've also managed, there's the imaging biomarker standardization initiative, which is of course a bit on the same side of what we're trying to achieve with PyRadiomics. And of course, we at PyRadiomics also joined in the IBSI in developing this standard. And some small differences remain, all of them in the pre-processing, but instead of just changing PyRadiomics in this instance, we've opted for documenting the differences, because these differences are all that it's not necessary that the one is better than the other, but there are two viable alternatives, but they're slightly different, like how you align a resampling grid. But in development, we also are adding replacement functions so that you can use PyRadiomics to generate fully IBSI compliant features. So in take-home messages with PyRadiomics, we've aimed to develop an open source Python package for Radiomics, which really takes you from image to feature, and then hopefully helps you to develop a very predictive model later on. It was developed with specific aim for generating a research community, with special care for readability of the code and also extensive documentation, helping in reproducible feature extraction through a single configuration file, but also the versioning of the software and the automated consistency checks. And finally, we really try to promote the standardization by simply by providing researchers with a ready-to-go implementation, which is mainly conformant with the now IBSI standard. So I would like to thank you all for the attention, and this here is our main developer team. But of course, the PyRadiomics community is growing, and we have had some contributions from lots of other researchers for which we are immensely grateful.
Thank you, Joost, for this very detailed overview of the example of a open source project. I see a question in the chat, so maybe we can start with that one. There was a question from Daria Malyarenko. She's asking about useful tools. Which approaches are you taking to ensure support and sustainability of your open source software?
So it's actually a very good question, because every open source software, well, any type of software, of course, needs to be maintained and further developed. And we at PyRadiomics, well, the main starting steps of getting this software up and running are now past, but we are still actively developing and really want to help researchers with requests for new features, but also maintaining the current functionality. And I think so that the main approach we implemented already quite early on is this continuous integration, so that any commit that we make gets tested by a quite extensive pipeline. So it tests whether the code still compiles successfully, but also if the results that are calculated are still compliant with the baseline we have defined. So the software actually implements also a simple CSV file that says, okay, if you use this configuration with this image, you need to have this value for this feature. And if it's not, then it simply fails and you can merge it with the master branch. And and really, of course, sometimes you do have developments that users actually do discover that there are issues and bugs and that features are calculated incorrectly. And in those cases, you can fix that, and then you have to actively update the baseline, of course. But that means that you only allow for changes in calculated feature outcomes if you are explicitly telling the baseline to update. So that means that you have consistency. And the other part is that we really try and promote also the usage in the community, and ask people that if they have a good idea to help us and share your ideas and codes.
Yeah, thank you. That's all very important, but I have a related question. So what about so that's a technical way of supporting the sustainability of your software, but what about the more like financial, if you want, or how do your colleagues from the core development times find time to work on this project? Is it a hobby initiative? Is it something which you can do part-time or full-time? How does it work?
So the main parts, so the main development of PyRadiomics, so the first 5 or 6 years, they were covered by an NCI grant, which is so the NCI has specific grants that aim at developing open source software. So they allow you to pay a group of researchers to develop this software with and they actually have the requirement for you to make it open source and report on it on the general meeting. We were there a couple of times with PyRadiomics as well. But for now, it's mainly also in part of, so PyRadiomics is still in that part where we are still the core development team working on it, getting paid for it, a bit less than we were at the outset, but enough to get at least the basic support there. But our aim is also to get, because people are using it and want to keep using it, and we aim to generate a community that really also makes time for it. So as people are using it and finding new and interesting ways to use it, that you create a community that really carries it. It's a bit, So an example, it's a bit like the 3D slicer program also. So it started out with a couple of grants that were written and used to first develop the 3D slicer, but although those grants all long gone now, so many people are using Slicer that there's actually a very active community of people who are developing Slicer also as part of their own research, because they are using it as part of their own research. And that's really what we aim to do, so to generate a community helping each other to keep this software going and preventing other users from really having to generate, to start from scratch with the implementing Radiomics features.
Yeah, thank you. That's very. I think that's a very good way to go. It's a very good wish, but it's also a lot of work to basically start the community and get it going, right?
Yeah.
Okay, I don't see further questions in the chat.
Maybe
Edwin,
I have
yeah.
a question. So as a clinical radiologist, I'm watching the developments in imaging biomarkers, and especially the radiomic biomarkers, with great interest. But there's always the question, how do you relate your radiomic outcomes to biological processes, right? How can you interpret what you're measuring from a clinical or biological perspective? And I'm just curious to hear your own thoughts, and what is your opinion on that?
Yeah, so of course it's actually it touches on the first and main hypothesis of radiomics, which is that of course different tumors will respond in different ways. So these are so the genotype, so you will, of the tumor will result in different outcomes, and the hypothesis of radiomics is that this genotype also results in a different phenotype. So how a tumor looks on the image is related to what type of tumor it is, and that again is related to the outcome. And it's a form of big data research that you first need to see if you can indeed correlate this phenotype to your outcome. And of course the second step would then of course be to see, okay, so what biological process is actually behind all this? So what biological process is the cause of this difference in outcome, and why does this also result in a different phenotype? And actually what you see that most research at the moment is mainly focused on this first step still. So first getting these models to work, so to find these correlations. And only limited research at the moment is really focusing on finding out why these correlations exist. So really the biological background. And from my point of view, it's a very important step, but at the moment I really don't have a valid answer for the question, because the old adage is also always the same: we need more research, but in this case, especially also in this area.
Well, I think that's very important. And again, your mission as a provider of the software is actually to enable this research. So I think it's really great. It's a very great initiative.